type rotors. In the name itself, we can found that a vertical angle rotors are going to have arrangement or holes for the centrifuge tube in the axis, along the axis of the central axis. So, these rotors are about 90 degree, that is going to be a angle of central axis. This is equivalent to the central axis angle, but the fixed angle rotors are going to have 14 to 40 degree. Here it is going to be 90 degree. So it is going to have a rotor speed which is decreased level than the fixed angle rotors, which is about 50 to 65,000 rpm. That means a revolution of these rotors are going to be 50,000 to 65,000 per minute. The capacity of the centrifuge tube is going to be 0.2 to 1 liter, which is equivalent to the fixed angle rotors. So here a small amount of wall effect to be occurs, but in general the wall effect is not prominent. Here the centrifugation is going to be occurs based on the biological particles based on the biological particles test tube. So the compound is going to be moved radially along the walls of the test tube, which is going to be on the surface of the test tube during the acceleration. When there is a deceleration to be occurs, that is when the centrifugation is going to be stopped, the compounds or biological materials to be involved in sedimentation, which moves on to the test tube surface radius along the wall. A small amount are going to orient within a shorter time to the pelleting out. So the small amount of wall effect to be occurs, but it was not prominent, because here the sedimentation is occurs because the compounds are going to move along the surface of the particle, but it was small amount of wall effect which causes the sliding down during deceleration. So the main advantage of this pelleting out is for DNA, RNA and lipoproteins, where it is going to have very fragile. So this centrifugation causes the sedimentation, which is not harsh wall effect to be occurs.